With that, I would like to welcome our speakers today who will be helping us explore the unintended consequences of these wetlands that you all came to learn about. They include Dr. Charlie Loewen, Assistant Professor of Ecology, Evolution, and Organismal Biology here at Iowa State University, as well as the graduate students who helped collect a lot of the data. These are Ecology and Evolutionary Biology Graduate Research Assistants, Daniel Edwards, Kaitlyn Sanchez, and Grace Jackson. So with that, we're going to head out to the field to learn a little bit more about this innovative research they're doing at these water quality wetlands. Thank you again for joining us.
So my name is Charlie Loewen. I'm an assistant professor in the Department of Ecology, Evolution, and Organismal Biology at Iowa State University. And we are out here today at one of our field sites for an INRC project looking at the co-benefits of nutrient reduction wetlands and whether or not they may have any sort of unintended consequences that we should be aware of as we are continuing to put these on the landscape. So what are some of the unintended consequences? Well, obviously, Iowa has a tremendous amount of agriculture, and so one of the consequences of that is following rain events and snow melts. A lot of the runoff and tile drainage that comes from these fields often has very, very elevated levels of nutrients. And another consequence of all this agriculture is that we drained many of the naturally occurring wetlands across the state. And so this creates a problem because wetlands are actually very, very efficient and good at reducing nitrogen loads and phosphorus loads sometimes before they reach downstream. And so some of the unintended consequences may be that, for one thing, they are much more efficient at reducing nitrogen loads than phosphorus. So we're fundamentally changing the ratios of some really key nutrients. And this is important because different organisms have different nutritional requirements. And certain organisms, such as cyanobacteria, which are a type of phytoplankton, or that live in the water column or attached to substrates, can sometimes produce their own nitrogen from the atmosphere. And so if we're alleviating some of the nitrogen pollution without having similar drops in phosphorus levels, we could be creating situations where cyanobacteria, some of which can produce harmful toxins, may be able to have a competitive advantage over other organisms, and it could possibly lead to more cyanobacteria downstream. It also is possible that just by reducing nitrogen levels overall and phosphorus to the extent that they do, we may have a drop in that, but we don't know until we look. So that's one of our goals, is just to see whether that is something we need to be concerned about going forward. So the Iowa Nutrient Research Center funded this project because restoring wetlands are one of our leading edge of field practices for reducing nutrient loads from agricultural fields. And so we wanted to go and look at, well, we're putting these things in the landscape. Are there other co-benefits for other biological values that we can try to maximize or optimize as we go forward and continue to create these wetlands or restore them on the landscape? So big part of one of our main objectives is to go out here and look at some of the lower trophic levels. So these are things like that fuel the broader food web, and we're looking at the health of those communities as an indicator of the quality of the habitat and the ability for these wetlands to provide useful habitat to water birds and other animals that we maybe want to provide more habitat to on these agricultural landscapes. One of the really valuable parts of field research programs, or just research in general, is having the opportunity to train students. So for this project, I was lucky enough to work with 3 graduate students who all are able to look at some of the biodiversity in these wetlands at different levels to focus on different communities. So with that, I'm going to turn that over to some of my graduate students to show you how they are sampling these communities to try to answer some of these questions we have about the functioning and biodiversity values of restored wetlands.
My name is Daniel Edwards. I'm a PhD student in the Loewen Lab. I am originally from Indiana, but I'm here in Iowa doing my PhD. I specifically study aquatic macroinvertebrates, which is this kind of big group of aquatic organisms that consist of a lot of different groups of insects, as well as some crustacea, some snails, and the likes. And this is a really interesting group to kind of work with because they've all kind of independently moved into these landscapes, and so they all kind of have these very different strategies to survive within these landscapes. And so because of this, different groups are more or less sensitive to types of environmental stress or change, different types of respiratory systems. If you imagine, a lot of different of these insects have gone from this terrestrial environment where they're breathing air to then colonizing the aquatic landscape where they're then breathing through gills or siphons. All these kind of different strategies allow different groups to being more or less sensitive to different types of stress in the environment. So these aquatic macroinvertebrates are really important for a lot of different functions. They do serve as this really interesting kind of aquatic terrestrial linkage. A lot of these organisms do not spend their entire life cycles in the aquatic environment, and so are actually exporting nutrients into the terrestrial landscape when they get eaten by birds or other organisms. They're also kind of this really nice kind of intermediate step between the aquatic like planktons and the invertebrates, and then they get eaten by fish or birds and other things. So they kind of are this really nice kind of middle level of the environment. My role in the research is to do the research. I'm out sampling the bugs. In these wetlands, I use several different strategies to do so. I sample macroinvertebrates upstream and downstream of the wetlands. And then within the wetlands, I use a novel strategy using this big trash can that I put into the wetland. And then within this, one of the interesting things that I'm looking at is this linkage between the plant communities in these wetlands and the aquatic macroinvertebrates. So how the different plant life and the different growth forms of those plants can kind of affect one another. And so I take this big trash can and I put it into the wetland, and this is my sampling unit. All the plants get taken out of this and then get rinsed off from the bugs. And then all the bugs I then take out of this with the net. We sampled once a month for 3 months. These communities change a lot over the year. So one of the things that we're thinking about doing in the future is looking at sampling year-round. Because these communities do change a lot as different organisms emerge out into their terrestrial life stage and then subsequently laterly colonize the wetlands again. I've always liked insects. The aquatic ones are a little bit more interesting because they're pretty generally understudied. They're super diverse. The aquatic beetles are probably my favorite. There's around 14,000 species worldwide, which is pretty crazy. And all of them kind of fill different niches and have really crazy adaptations to survive in the aquatic landscape. That's just really fun and really cool.
Hi, my name is Kaitlyn Kelly. I'm a member of Dr. Charlie Loewen's lab. I'm a master's student. I'm a first year. I'm initially from Southwest Iowa, and I was interested in the project going on up here at Iowa State, and so I became a member of the lab. My primary role in the lab is to look at the zooplankton communities, and so as our project looks over a bunch of different communities within these wetlands, I am specifically looking at zooplankton, which are microscopic invertebrates, and they are primary consumers of phytoplankton and algae, and so therefore it's important to look at them and what their communities are doing in terms of these wetlands and in terms of food webs, because they are important roles in the food webs and they provide food for the macroinvertebrates and the larger vertebrates such as fish and other things living in the wetlands. Zooplankton provide food for both macroinvertebrates like different bugs, and then they also provide food for other vertebrates such as birds and amphibians and other reptiles that might be living in the wetlands. So I have a couple different sampling techniques, but I have one that's more interesting that I'll show you today. And so that will be using a vertical Van Dorn, which is basically this clear tubing, and it has plungers on both ends. So you'll open both ends of the plungers. You'll send a messenger weight down, and it will hit a button where it will close those plungers. And so traditionally, the Van Dorn will be placed in the water column at this time. And then once you're at a specific depth, you'll send the messenger down. It'll close the plungers, and it will trap the water where the Van Dorn is sitting at that moment. So I've always been interested in anything aquatic. And so I was working a position full-time for the Crumpton Lab before I became a master's student. And so I was already quantifying zooplankton for their ambient lake monitoring project. And so I was really interested in also looking at zooplankton communities within wetlands rather than lakes, because lakes have been very traditionally looked at in terms of zooplankton communities. So it was something new to learn about.
Hi, my name is Grace Jackson. I'm originally from Dayton, Ohio, and I moved here 2 years ago to obtain my master's degree in the Loewen Lab. My role in the lab is studying the base of the aquatic food web, which is phytoplankton and periphyton. Now, these two groups are sort of like plant-like organisms, but they include a wide range of life, including bacteria and protozoa. And they have the ability to photosynthesize. So they take energy from the sun and convert it so that other organisms, such as the zooplankton that Kaitlyn talked to you about, and eventually up the food web into the macroinvertebrates and the amphibians, can obtain that energy too. Phytoplankton, phyto meaning obtaining energy from the sun, and then plankton meaning they're floating freely in the water. So that is one group that I study. That's the group that forms the algae blooms that you may have seen around the state as a result of the nutrients coming in, because phytoplankton use nitrogen and phosphorus as well. And then periphyton are just a different life strategy that these plant-like organisms can use, where they attach to a substrate, such as a rock, and they grow almost like a microscopic forest structure. So I'm studying those two groups. I measure phytoplankton similarly to how Kaitlyn samples her zooplankton, basically by taking a surface grab of water, and then we use a special instrument called a flow cam, which is basically a fancy camera and microscope. You put your water sample in at the top. It goes through this microfluidic tube, and then there's an objective that's positioned right next to the microfluidic tube, and it captures tons of images of these microscopic plankton coming through. And then I can take those images and actually train the computer to recognize certain shapes, and it will help me sort these different groups of phytoplankton, and then I identify them just based off of traditional methods from that point. One of the objectives of this project is to understand how this wetland is affecting the biological communities up and downstream. We looked at periphyton, which again is the attached algae, using these special samplers. It's basically a flotation device that carries 16 microscope slides. We tie this device to a cinder block with some string, and we deploy it, and it floats on the water's surface or just below the water surface for about a month, and we come back, and there's a bunch of periphyton that have grown on these slides. And then we can take those slides back to the lab for analysis, either using the flow cam, or we've also frozen them for pigment analysis. And then we can do pigment analysis in the field using a handheld spectrophotometer. And basically what that is it has a light source and a lens, and you put it to the microscope slide that has a bunch of periphyton that's grown on it, and it flashes, and then it gets a reading back, and whatever the reflectance back from the periphyton is, every kind of algae has its own special amount and types of pigments, and there's a computer in this handheld spectrophotometer that recognizes those particular ratios and types of pigments in order to identify three main groups of the periphyton, which is cyanobacteria, diatoms, and green algae. One of the big questions when we started studying these wetlands is there more cyanobacterial growth before or after the wetland, or is there really no difference? And the reason why we were interested in that is because certain species of cyanobacteria are known to be what is known as nuisance species, because they can produce toxins that affect our water quality and our public health.
So that largely concludes the outdoor portion of this, where we've kind of demonstrated how we are sampling some of these wetlands. Again, our objectives here are to understand the biodiversity within these restored wetlands so that we can, hopefully as we continue developing them on the landscape, perhaps use some of our learnings to build them in ways that may further optimize some of those biological values, as well as avoiding any potential problems if we find them. Very exciting as we continue to build these on the landscape to help improve our downstream water quality, and again, hopefully try to associate increasingly additional values for biodiversity, things like water birds, to help them on these very agricultural landscapes.
All right, so at this point, folks can join me in the conversation. If you have questions for our presenters today, feel free to submit those in the chat. We will get to those here. One of the questions I had was, regarding how many sampling locations you were visiting, how many different wetlands?
Yeah. We got out to 9 different sites. And then we also looked at a couple of additional, CREP sites are these Conservation Reserve Enhancement Program websites, which were designed for water quality improvement. So these are some of these restored wetlands that we're focusing on. And then we also went out to 3 different sites that were more for comparison sake. One of them actually is a remnant wetland, so it's been there for ages. Another one that has been, or another couple that have been restored. A few decades ago that are more designed for wild life habitat specifically, just for comparison points.
And so now maybe open it up to the graduate students if in your first year or early preliminary data, based on those different wetlands, were you seeing any differences, um, good, bad, or otherwise, as you compare how those are designed, built, and the age
Sure.
on your results? Maybe that's too many variables to talk about all at once, but
Yeah, I'll let the students go through it one by one here, but I just want to start by saying, these they're all working very hard, and as we kind of saw, a lot of these critters and phytoplankton are very small. It takes some time to count, so we are still finalizing some of that initial data analysis. So any sort of results we may have are still very early stages and preliminary, but I'll welcome them to kind of talk about what they've been seeing so far. So, Daniel, you want to start?
Sure. Yeah, I can start. So I have noticed some pretty clear kind of trends generally with the different wetlands. One of the biggest things that the aquatic insects are associated with are those kind of macrophyte communities. And so the biggest distinction between them being where there is emergent vegetation or submergent vegetation. And one of these things that varies the most between the different wetlands is the proportions of emergent to submergent vegetation. Generally, the emergent vegetation is associated with higher diversity of aquatic insects. A lot of the insects use it for respiration. They actually plug into those plants to breathe. Other ones use them to they crawl out and need those emergent vegetation as a structural component to be able to emerge out from their larva to an adult. They need to be able to perch on a cattail leaf to be able to dry their wings before they fly. And if they don't have that, they can't reach adulthood. And so that's one of the big things that we can kind of see is that emergent vegetation being an important factor in some of this. But yeah, I can pass it on to Grace.
Yeah, so it's really hard for me to say any kind of trends. My eyes get kind of blurry when looking at the hundreds of thousands of algae images. Um, but I will say I have noticed a lot of diatoms in the paraphytic community. So that's the algae that grows attached to a surface, in this case, my slides. And it's been really cool to see up close some of the adaptations that they have to attach these surfaces. So diatoms in particular can produce these mucilaginous stalks. It's like these little legs that come off and they grab onto the surface. So that has been really cool. But yeah, I don't have any specific trends, but I will be presenting at a conference at the Association for the Sciences of Limnology and Oceanography in May. So we'll hopefully have more to talk about then. But yeah.
Hi, I also don't have too many data trends, at least per wetland right now, but I have seen kind of overall throughout all of the wetlands, I have been seeing a higher taxonomic diversity within kind of like the zones that Daniel mentioned. So the zones with the macrophytes that are emerging out of the water, and then the macrophytes that are submerged within the water. So I'm seeing a lot more diversity within those zones rather than out in the open water. And this could be due to a number of reasons, but a couple of those could be just coverage from predators. So macroinvertebrates and fish predate on zooplankton. And so plants just in general can provide some cover from those. And then as well as just feeding behaviors in general. So a lot of the large-bodied zooplankton, like you guys saw on the video of the scope, so those will graze on periphyton that the periphyton that Grace is looking at that is growing within or on those macrophyte plants. And so they're just more likely to be in those zones as well. And then also I have seen a bit of a difference in the community sizes and just the amount of zooplankton that are there, and then a lot lower diversity, just depending on the sampling month. And so this could also be due to a bunch of different reasons, but for zooplankton, I would say generational trends. So just the turnover of their different generations as they grow and then die and then begin to replace those communities. So that's about all I have so far, but I'm also working to further analyze the data and find some trends based off of functional ecology and response to the nutrient gradients as well as the different vegetational zones.
Yeah, so speaking of nutrients, is there data being collected to evaluate the concentration of nitrogen and phosphorus at these sites? And then how do those maybe, they're curious what those typical concentrations were this past summer, if you know offhand.
Yeah, so we did collect that, and we're also piggybacking off some other work in these same wetlands where they have auto samplers, some more finer resolution data. So that's going to be able to leverage that, hopefully try to explain some of these patterns. But yeah, the values vary tremendously, especially after sort of rain events and things like that. You often see these big spikes because we're just measuring like ambient concentrations at that time. So yeah, a ton of variation across that. Most of these are very like eutrophic to hyper-eutrophic systems, not so dissimilar from most other surface waters in central Iowa. And I will just add a little bit briefly off some of what my students mentioned. We are finding a fair bit of diversity in these systems, which is really great to see. We're still, and a lot of variation as well. So a lot of our next steps are going to be trying to explain that variation, why we see more variation, or sorry, more diversity in certain places than others. And the same thing with some of the cyanobacterial questions. We are finding cyanobacteria, sometimes more downstream, a couple times more upstream. And so we're still trying to explain those patterns. So that's going to be our next steps there. But yeah, relating it to nutrients is definitely a big objective.
Yeah. So you caught that question in there. Yeah, trends in cyanobacteria, that definitely perks Bill's ears up because of the negative connotations with that, understandably.
For sure.
So let's see here. You talked about some of the different plant species you see out there. So there's a couple questions. I'm going to maybe kind of group them together, but problems with excessive duckweeds or other invasives impacting different sites and how that might impact your data collection?
Yeah. I'll let Daniel talk a little bit about that. But yeah, duckweed is very, very common as a macrophyte. That was one of the things Daniel was looking at. Some of these sites, especially towards the end of the summer, kind of get fully covered with them. So that's definitely going to affect, the availability for light to penetrate into the water column to be, support up. But Daniel, do you have any thoughts on duckweed you want to share?
Yeah, like one of the things that I was kind of sampling is any of the plants that are kind of, if their feet are in the water, I'm kind of interested in them. And so it was kind of interesting to look at some of these different invasive species. I think ultimately I found only a handful of them. Some sites were a lot better than others. But like curly pondweed was at least at like 2 or 3 sites that we were at. One of the biggest ones is the reed canary grass. We saw that at a lot of sites. Probably over half of them were pretty well dominated by reed canary in some area of the wetland. But the duckweed is an interesting one because it's not necessarily non-native, but it does have these tendencies to kind of really disrupt the ecosystem. And there are some areas where it would be like several inches thick of duckweed from the surface of the water down. And I haven't really looked at what exact effects it's having on the aquatic insects yet, but I do have that data and I'm really interested to look at it. But yeah.
It's likely also to some extent affecting some of the oxygen patterns we're seeing. We find some of these systems really get quite anoxic, especially down at depth. And so when you have less ability for light to penetrate into the water column, you may have less photosynthesis in the water column and greater potential for having more anoxic or oxygen poor conditions as well. So it may also be influencing that. And again, these are some of the patterns we're planning to explore further.
Yeah, so kind of tying back into that earlier question, um, in terms of the 9 different wetlands, were they all constructed wetlands or some of them natural wetlands? And then how would they rank in terms of permanent to semi-permanent water pool?
Yeah, so the 9 core study sites were all restored under this CREP program. So they're all on the Des Moines Lobe, and were designed to basically receive tile drainage and provide treatment. So they're all kind of similar in that regard. They were developed. There's a bit of a range in how in the ages of them, but they all have a very similar design where they basically put a weir up to create this pool. So for the most part, correct me if I'm wrong, guys, but I think that like they were all standing water in those pools the entire time. The weir helps accomplish that, right? So provide sort of a pretty stable flow environment for the most part. Well, obviously, when after a rain event, you get more flow and you get more runoff over top of the weir, but the weir helps kind of stabilize the water level.
Would you say land use variation in the watersheds that are contributing to these 9 wetlands are fairly similar in management?
Yeah, I mean, so they're all basically receiving tile drainage from agriculture. Being the Des Moines Lobe, the vast majority of that is corn, right? So there's going to be some variation probably in how these fields are managed. That isn't something we've really dug into yet. We've just been focusing on what's in the water specifically. But that would be another kind of interesting way to take it if we can, if we see some patterns, if we can relate that to trends in the landscape as well, that would obviously be very interesting as well.
Yeah, so maybe this is some question. I think, Grace, if I can track. Oh, yeah. Because you talked about I think the field fluorometric, if I say that correctly.
Yep. Yeah, the
Uh,
spectrophotometer.
yeah.
Yeah.
Do you use that to evaluate general phytoplankton groups, is the question.
Yes. So yeah, it's the general periphyton groups, and it's greens, diatoms, and cyanobacteria. So 3 big groups, and it just gives you like a ratio of that little space that you've scanned. Um, well, I guess I'm calculating a ratio from it. It gives you like a readout of like how much, I think it's like per like millimeter or something like that. But yeah. Yeah, so just those 3 main groups.
Yeah, it allows us to estimate sort of a biomass of the different groups, as long as you don't have too much thickness in the sample. If you have really thick communities, it has a hard time figuring out that the depth of them. But it's very good for kind of area type measurements. They do have similar devices to use in the water column as well for the phytoplankton, but we have this device to look at the attached communities.
So, yeah, there's a question. I'm trying to make sure I phrase this correctly. So will the nutrients be measured directly to gauge loading and export and or help with biological sampling so you can create some sort of indices if you see these certain communities at these levels, kind of giving you a sense of how it's functioning?
Yeah, yeah. So we're not as worried about loading as much in this as sort of like what the concentrations are at a moment in time. Loading implies like how much the concentration is going with how much flow and like how many it's delivering nutrients downstream. We're more focused on like what's happening in the wetlands and immediately downstream, which is more of a concentration than a loading question quite often. But yeah, for sure. Like part of the reason these sites were selected is that they do span a bit of a gradient in terms of nutrients as well as the relative amounts of nitrogen and phosphorus. So getting an idea of ecosystem health under these different conditions is definitely one of the objectives here. Both in terms of just sort of understanding what the co-benefits of biodiversity, and again, if we see situations which are less ideal, where we maybe have more cyanobacteria or less diversity generally, if we can kind of identify, what types of wetlands we're seeing those in and what sort of water quality contributes to that, that'll also be helpful to hopefully try to avoid those situations in the future.
Trying to keep up with the questions coming in, so I apologize if I missed somebody's question. Uh, but kind of taking a step back, in general, what role does aquatic life play in reducing and or releasing nutrients to the water?
Yeah, so um, nutrients, fuel production. So the phytoplankton, the periphyton, they're going to take that up out of the water column. They're going to convert that into biomass. They're going to be eaten by other things. So kind of, I think Daniel touched on that a little bit in the video about how sometimes these wetlands can also be sources of nutrients when you have like aquatic insects and things like that, growing in the aquatic system and then leaving for part of their life stage. So they can kind of have this in and out flux of nutrients. That's probably the main way they're going to reduce them. Otherwise, there's denitrification, right? Which is the main, yeah, I guess I probably should touch on that. So one of the main reasons that these things are good at reducing nitrogen loads in particular is through natural denitrification processes, which tends to happen primarily down in the sediments. So that is sort of why these things are useful for this. That's sort of why we are using them as edge of field management practices, is because they have the ability to take nitrogen in the water column and basically release it into the atmosphere. With phosphorus, there's no atmospheric form like that. So that's why they're generally not as great sometimes at reducing phosphorus, because it can't take it anywhere. That phosphorus may fall into the sediment and can sometimes be buried, and that can sometimes be a long-term solution, but also big storm events, you have a lot of flow, can sometimes stir that back up and release it again. And also under anoxic conditions where there's really low oxygen, it can sometimes be internally cycled as well. So those are some of the general nutrient patterns. And then yeah, we're looking at sort of how a lot of the biodiversity is like responding to some of those differences in nutrient availability.
There's a question here, and I will not butcher the type of the bloom, but have you noticed any development of the blooms in which the color of the surface water is deep red? They've been reported in warm, shallow, stagnant, nutrient-rich bodies.
It's really interesting. I did not see any red blooms across the summer. But that's happening in Iowa, you guys? This
It
person
can
is?
happen. I'm aware
Okay.
of another student working in Crumpton's lab who brought a sample with some red algae that he was interested in. So we didn't notice that while we were sampling it, but it's certainly possible.
It's really interesting. I'll have to
Yeah,
ask them
yeah,
about
for
that.
sure.
Well, and I will note that in 2025, we had adequate moisture, that these wetlands were definitely not stagnant. There was continual flow. So maybe that was also a reason why you wouldn't have captured it last year, if it really
Yeah.
does need that stagnant water aspect.
It may well, and the nutrients that change with that and the oxygen availability, all these things will contribute to what's there. And then, yeah, in that summer that we did the sampling, we did have a number of good rain events. We lost a sampler here or there. Things like that happened to some of the flooding. So in the Northwest, I think there was some flooding that year.
Yeah, I can imagine that your little floater could have taken off no matter
There's
how
a couple of
tough
those
that
that,
knot.
yeah, moved their way downstream a little bit. Yeah.
Yeah. So is there any interest or potential for comparing, say, these CREP wetlands, so these Conservation Reserve Enhancement Wetlands, spell out that acronym, or these water quality enhancement wetlands that are constructed to receive tile drainage for the main purpose of nutrient reduction, compared to some of the historic or restored wetlands that function a little bit differently on the landscape while still providing some of the nutrient reduction?
Yeah, and that's really one of the driving questions, is to kind of see what other kind of co-benefits they provide biologically, right? So, as I mentioned, we're finding a fair bit of diversity in a lot of these systems, often comparable to some of the sites. Like we only had one remnant site. It's actually not that easy to find comparable remnant sites in central Iowa, but as well as some older restored wetlands that were designed more for providing wildlife habitat. We are often, like, we're still going to make that comparison more formal, but just sort of squinting at the data, we're seeing a lot of similar taxa. And so that's a good sign that hopefully it is able to provide some of that habitat and support some of those food webs. But yeah, it's still just kind of very early looking at that data.
And kind of larger picture, how might this research here maybe inform future designs of these water quality enhancement wetlands?
Yeah, so that's really what we're hoping to get to as well, right? So where we see, like I mentioned, there's a ton of variation in the diversity, in the cyanobacteria, and all these different things. So attributing some of the causal factors to those patterns is really, the goal here, right? And so if we can find things, as Daniel mentioned, where we have more variation in different types of vegetation communities, we're typically seeing more variation in the insect communities as well, which is generally a good thing for supporting higher trophic levels as well. So Relating to design elements where we can, so kind of looking at the bathymetry, certainly obviously the nutrient inputs, which we only have so much control over, the tile drains with the tile drains, but where we can kind of see definitely encouraging a wider variety of vegetation communities seems to be an important thing to do, which isn't always considered. Sometimes they just sort of, put these there and let them just naturally colonize, but there may be, if we can find certain types of plants that facilitate better outcomes, then that would be a possibility to possibly try to seed those sort of plants and encourage the colonization. So things like that, I think, are definitely important. Anybody else have any thoughts that I'm missing on that, Kaitlyn or Grace or Daniel? No? Okay. Those
No,
are
I
those
feel
are the main
like,
things.
yeah, I feel like the biggest thing that at least Caitlin and Daniel are finding so far is just the importance of the emergent zone.
Yeah.
Yeah.
And so
Yeah,
all
good.
these microscopic creatures and some of these bugs, they're finding their way to these wetlands. It's not like they were inoculated or brought into the system. So it's kind of if you build it, they will come situation. So what can
Mhm.
we do to help build it up for them to be as, healthy and beneficial as possible?
For sure. And I'll folks can talk about their individual taxa, but just in a general sense, a lot of these things are quite widely dispersed. Some of the plankton in particular, they can disperse widely in the wind. They can attach to waterfowl. Even a lot of like zooplankton can survive desiccation. A lot of them can be survive being digested by waterfowl and actually come out in the feces and use that as a colonization event. So they have a number of really kind of spectacular strategies to do that, but some organisms are better at that than others. So I think that there will be certain groups of organisms that can potentially benefit more from those sort of more intentional efforts. Something like zooplankton and phytoplankton in a place like Iowa probably doesn't need as much of it because they're pretty strong dispersers on their own. Maybe some of these more dispersed eliminated insects or macrophytes maybe could use more help at times. But yeah, if anybody else has anything to add to that.
Yeah, I can add a little
Yeah,
bit more on
no,
the
you go
insect
ahead.
side. Um, I think it varies by taxa,
Mhm.
very obviously. Some of the dragonflies I'm finding are actually migratory species. And so some of these wetlands are like stopover points where they can complete their life cycle before kind of continuing on this big migration path between Canada and going back down south to like Mexico. But then other taxa, they only maybe fly like a mile at most. And so that connectivity of habitat, of maintaining aquatic habitat so they can kind of jump between pond and pond can be important for those taxa. So
Um, I was just gonna add, Charlie talked a lot about how zooplankton are able to desiccate and yeah, they can basically their eggs can dry out and be stored in the sediments, and then of course that can also be distributed during like flooding events and stuff like that. And not related to my taxa, but I remember learning in a class that there are some algae that can desiccate and they'll fly in the wind, and some of them are even found in bird baths. So just very easy for them to get taken up by the wind and just find a source of water and end up being able to thrive there.
As you've gone out to these different sites, have you had the opportunity to interact with the landowners? Because these are privately held wetlands. They are not public spaces. But obviously many of them are willing to have research conducted on them because they believe in what these practices can do. But if you have interacted with any of them, what has come out of those conversations?
Yeah, we were really grateful to the landowners. I feel like they were really welcoming. Most of them we didn't see, but there was a few that would come up and talk to us, and yeah, they were interested in our work, and a lot of them appreciate and enjoy the birds and other animals that call their wetland home. So, um, yeah, honestly, nothing but positive interactions.
Yeah, there was a comment early on after we finished the video that all presentations would be better if there were birds singing in the background. And I can't disagree with that. So, folks, if you still have some questions, feel free to get those in. I'm going to share my screen and one more time for those that joined us today for CCA CEUs. Please be sure that you email me. My email is there on the screen and I'll drop it in the chat here momentarily. But please provide your name, the name you used to watch today's virtual field day, if it's different than the name you have registered with the CCA organization, and then your CCA number, and I'll get those submitted to you for credit tomorrow. And once again, as a USDA-funded program, we do ask our participants to please complete a voluntary demographic survey. You only need to do this once, so if you've already done it for a virtual field day or webinar in 2026, thank you. But if you haven't, you can access it either by quickly scanning that QR code. I'll leave it up on the screen here in a moment, or visiting iowalearningfarms.org/survey. Really appreciate you doing that for us. So thank you. And let's see. Come back here. Ooh, so I'm not sure on the second one. We did touch on duckweed, but filamentous algae. There's not necessarily a question on this, but if you want to touch anything on what you're finding or whether that's good or bad.
Yeah, I've definitely seen some filamentous algae. Um, Whether it's good or bad sort of depends. So some of them produce toxins, some of them don't. It depends on how dominant they are. Um, but some of them are like uh, harder to eat than others in regards to the like zooplankton that are eating them. But yeah, I've seen it. I don't have a specific like trend to talk about filaments, but that is something that I am looking at as
Any
well.
maybe
So,
correlation with the duckweed, or is that pretty prevalent at all your sites?
oh, that's a interesting. I hadn't thought about looking at the
When
correlation. Yeah.
Yeah, at least
Go
when
ahead.
I see a lot of like duckweed dominance, I don't see as much of the filamentous algae. At least just like anecdotally, as I'm out there, it seems like it's either one or the other.
I would
Likely,
agree with that.
yeah,
Like
likely
that's
one
like
is
It,
just becomes dominant and the other one can't. But
it's important to remember a lot of these things are competing with one another, right? And so they compete with nutrients, they compete with various things. One of the things they compete for is light. So if you have a very, very thick surface of duckweed, it's going to be harder for a lot of other photosynthesizing organisms to establish kind of below that canopy, right? If you can imagine a forest canopy that's really, really thick, a new tree has a hard time establishing until one of the trees fall down and then you have a hole in the canopy. So it's sort of a similar dynamic in these wetlands.
And in most cases, that duckweed came in as a hitchhiker. It was not part of the seeding mix that would have been
Yeah,
used to establish the vegetation there.
duckweed can get around pretty well. Yeah.
All right. Well, thank you so much, everyone, for tuning in today. If you have additional questions, you can find contact information on the Iowa State website for this lab and for our speakers here. But I will turn things over for any final comments from each of you. And again, thank you to the Iowa Nutrient Research Center for your support of this research and today's virtual field day.
Yeah, I guess in terms of final comments, yeah, thanks everybody for tuning in today. Kind of wish we had a bit more firmer results to show you, but please, kind of those will come. But we'll hope yeah, over the coming months here. Trying to get an idea of again how we can just especially as we continue building these things on the landscape, how we can manage them, how we can build them to try to optimize these benefits, and where if we do find any kind of problematic situations, how we can kind of mitigate those. Not that I'm saying we're seeing positive things, but you don't know unless you look, right? So we're doing some original kind of looking into these potential issues, and yeah, results will be forthcoming.
All right. Well, thank you, everyone, and I wish you all a fantastic afternoon. Take care.
Thanks, everybody.
Thank you.